Welcome to ASH News TV. I'm Rachel KOPCZYK.
And I'm Ed Hyland. The FDA recently approved 2 new drugs for the treatment of AML and brought back 2 old ones.
The ASH and FDA Joint Session discussed their safety and efficacy for patients. Product reviewers from the U.S. Food and Drug Administration, which co-sponsored this session, discussed their safety based on clinical trials and toxicity studies. Clinicians also offered their perspectives on the use of the products in the real world.
Enasidenib was granted regular approval for the treatment of adults with relapsed or refractory AML with an IDH2 mutation as detected by an FDA-approved test. The decision to grant regular approval on the basis of a small, single-arm study was context-dependent. IDH2 mutated AML is a very rare subtype of a rare cancer that represents a high unmet medical need.
Remarkable progress has occurred in our understanding of acute myeloid leukemia, and we've begun to realize the progress of decades of work as evidenced by the new approvals this year. It feels to me like we've finally found our oasis in the desert of acute myeloid leukemia.
There were 2 featured topic discussions at this year's annual meeting. The first looked at the utilization of drug combination therapies, and the second discussed the hemophilia revolution currently underway. The advances in hemophilia care have been significant, but substantial challenges remain. For example, neutralizing antibody formation, particularly for patients receiving factor VIII infusions. The research presented in this abstract applies to a wide range of bleeding disorders.
So we have coagulation inducing thrombin formation and fibrin formation, but really to tamper that, we have inhibitors including tissue factor pathway inhibitor and antithrombin. And so we normally have a balance, but without factor VIII or factor IX, you then have bleeding. And then if we can inhibit tissue factor pathway inhibitor, and tissue factor pathway inhibitor inhibits the tissue factor 7A, 10A complex. And if we can inhibit that, then we should be able to restore normal or close to normal hemostasis.
This featured topic discussion focused on rational combinations of targeted agents in diverse hematologic malignancies.
The most interesting thing was that there is now a tremendous interest in the concept of combining novel agents with each other in a rational fashion that is designed to overcome the mechanisms by which tumor cells, cancer cells, develop resistance to a particular targeted agent. The concept is to circumvent that problem by disabling cytoprotective pathways that allow the tumor cell to escape the initial targeted agent.